Okay, so we've got essentially two different types of movement of materials across the plasma membrane. We've been talking about passive process, so passive diffusion or passive movement, meaning that no energy is involved. Okay, so the cells just move via chemical reactions and chemical doings to cross the membrane. But sometimes molecules are large or being moved strongly enough against their concentration gradient that the cell needs to apply a little bit of energy in order to get that molecule across the membrane. So that's called active movement or an active process. It's going to require ATP. Okay, so passive is when molecules move naturally down the concentration gradient. Just when things dissolve and maintain their spatial distance naturally. So molecules naturally want to move out and be as spread out and equal as possible. Okay. Diffusion has many different factors that are going to influence the speed. Okay. The steepness of the concentration gradient. So if you have 100 on one thing and nothing on the other, they're quickly going to move in order to make it 50-50. Temperature, the warmer it is, the faster molecules move. So the faster we're going to maintain or get to that equalization point. Size and mass, the larger it is, the slower it moves. So tiny things are going to equilibralize a lot faster than large things. Surface area, okay, and the distance. So if your plasma membrane is really thick, it's going to move through a lot slower than really smaller, narrow plasma membranes. Okay, we talked about some of the passive processes, but another one besides diffusion is osmosis. Osmosis is the movement of water. The movement of water through this selectively permeable membrane. So water is just large enough that it cannot move through the lipid bilayer on its own. It needs some of these membrane proteins in order to move it, but it will move naturally down the concentration gradient. Okay. It's going to do this in order to maintain equilibrium. Okay, so we want just enough solution in order to maintain what we call an isotonic solution. So it's naturally going to want the same amount of water on the inside as the outside of the cell. So when it's totally equal, we call it isotonic. Okay? If we have more on the inside than the outside, okay, more water on the outside, more solutes on the inside, that water is going to rush in and burst the cells. Okay, so hypo, I always think of hypo as exploding. Okay, you can think of a balloon popping. Okay, so hypotonic has more water on the outside of the cell, causing the water to rush in and swell and burst the cell. Hypertonic solutions have more water on the inside of the cell than the outside environment. So left up to natural being, that cell is going to shrink as all of the water leaves the cell in order to maintain the equilibrium and get more water on the outside. Okay, so hyper causes the cell to shrink up. We're looking at examples of these are red blood cells in different solutions. So if your blood isn't properly solubilized, then it can cause the red blood cells to either rupture or shrink up and shrivel if the electrolytes and the fluidity of your blood gets off balance. Okay, so one of the other types of passive is facilitated diffusion. In facilitated diffusion, molecules move across the membrane via transporters or channels, but they are restricting the particular molecule, but they still are moving naturally down the concentration gradient. Okay, So it requires a carrier protein in order to allow some of these larger materials in. So we have two different types of facilitated diffusion. We can either have carrier mediated, which is when you've got a protein that is specific to carrying one lipid or chemical. It's going to allow that molecule to bind to that membrane protein before the shape will change, and it'll kick it through according to concentration gradient. Or we have channel-mediated facilitated diffusion, where it just allows a lot of different small materials to go through. Okay, we've talked about all of the passive diffusion, the things that go with the concentration gradient and do not require any energy. Now we're going to talk about active transport. We've got a couple of different types of active transport, but the big thing to remember is that they all require energy of some sort. Okay, primary active transport requires energy from ATP. It is going to do a movement or a pump to move materials across the concentration gradient. Okay, so instead of going from high to low, it's going to go from low to high. It's going to have a much more complicated membrane protein that is going to require this energy in the form of ATP. So ATP is going to release the energy in the form of breaking off one of these phosphate groups, making ADP an energy released, and that energy release is going to shove, in this case, the sodium across the concentration gradient to the extracellular matrix. Another type of active process we have is secondary active transport, and this is going to use energy that is stored in ionic concentration gradient. So we talked about having ionic concentration gradients making the part of the cell positively or negatively changed, and it's going to be through that charge that we're going to be able to pass some materials. Okay, so it indirectly uses the energy. It requires energy to get this charge through the primary active transport, but then it can utilize it again. Okay, so we've got primary active transport here utilizing ATP to get sodium outside of the cell, causing this charge, and then it can use secondary active transport in order to move the sodium across the concentration gradient, because we've kicked out 3 sodiums here and we're going to allow 1 sodium back in. With the process of allowing that sodium to move in, we can sneak through some glucose as well. Okay, we've talked about the plasma membrane, which is the exterior part of the cell. Let's go on and talk about the cytoplasm. The cytoplasm is all of the goo, as I like to call it, on the inside of the cell. So you can think of it kind of like Jell-O. It's not, but it's a good way to think about it. Okay, it's located between the plasma membrane and the nucleus. So it's all of the stuff within the cell other than the nucleus. We have in the cytoplasm cytosol. Cytosol is mostly water, and within that water it's got lots of solutes dissolved in it. Those solutes can be proteins, salts, sugars, those sort of things. We have cytoplasmic organelles, so organelles that float around in the cytoplasm, and these are the metabolic machinery of the cell, so it's what keeps the cell going. And then we also have inclusions. Inclusions are granules of glycogen or pigments, lipid droplets, vacuoles, and crystals, all kinds of other things that are floating around within the cytoplasm that help with cellular functions. Okay, before we get going on organelles too much, I wanted to take a second to talk about ways that we get materials in and out of the cell. So in this case, we're going to be talking about this is the plasma membrane. Okay, so this is that phospholipid bilayer. So we can do what is called endocytosis. Okay, we're going to bring things inside the cell through active transport, through cellular enveloping. Okay, so endocytosis is essentially taking the plasma membrane, reaching out, encapsulating something, depending on what it is, depends on the type of endocytosis, and then the cellular membrane will then pinch off and bloop that material inside the cell, depending on what it's gathering and collecting. So the first thing we've got is phagocytosis. We go through phagocytosis in order to take up large particles. A type of cell that is really good at going through phagocytosis are amoebas. Another thing is some of our white blood cells roll around and they go through phagocytosis in order to take up unwanted materials that are floating around our blood. And typically in that case, it's going to be things like E. coli or foreign bodies or bacteria. They're going to roll around and they're just going to eat all of this stuff and take it into the cell, and then they'll break it down. Okay. We can also go through a type of endocytosis called pinocytosis. Pinocytosis is taking up small particles in fluid. So it's drinking in fluid and particles and making these little carrier containers inside the cell. Okay. The last type of endocytosis is receptor-mediated endocytosis. So sometimes we need to selectively bring materials in, specific ligands into the cell, and that can be done through receptor-mediated endocytosis. Okay, just like we can bring materials into the cell, sometimes we need to remove things from the cell. Okay, so when we're kicking things out of the cell, it's called exocytosis. Okay, so it's essentially the opposite of endocytosis, but it is a way of exporting material out of the cell. This could be material that the cell made, it could be waste material, it all kind of depends. Okay, let's talk about the organelles more specifically. So organelles you can think of organs. We all know what organs are. We've got hearts and lungs and kidneys and those sort of things. Different parts of the body that function to make the organism work as a whole. At the cellular level, we have organelles, little organs, that perform functions to grow, maintain, and reproduce the cell. You guys covered a lot of this in the lab in week 3, so hopefully this is all a review. Okay. Here is a generalized photo of a cell. So on the outside, we've got the plasma membrane. On the inside, we have a giant enclosed ball that we call the nucleus. It's enclosed by the nuclear envelope.